I said we're just going to give everybody a couple more minutes because I know some people may be coming from another meeting. But just as a reminder, this week is our graduate student invited speaker week. So the graduate students will be monitoring the chat for the question and answer. But we're so thankful for Dr. Usselman to be here today. I know a lot of us are really excited for your presentation. So thanks for coming.
Thanks for having me. I'm very excited to be here. You asked me back in December and I was like, I'm going to put together a new presentation that I've never given, which is the classic thing to think in December when it's 3 months away. And then this week I was like, why did I say I put together a new presentation? But it was very fun. I'm very happy that I did it.
Now you'll have it for future use.
Yeah, exactly. You guys can be my guinea pigs.
All right, Dane, we could probably get started.
Perfect. Yeah, so as was said previously, this is another graduate student chosen speaker. I just want to thank you all for attending, and it is my honor to announce our seminar speaker this afternoon, Dr. Charlotte Usselman. Briefly, she completed her master's and PhD at Western University in London, Ontario with Dr. Kevin Shoemaker, then went on to do 2 separate postdocs, one with Dr. Steinbeck in Davenport at the University of Alberta, and a separate postdoc with Dr. Nina Stockenfeld at Yale University. Her training has led her to become a truly integrative physiologist with expertise in both cardiovascular and neurovascular measurements, just to name a few. And she's gonna kind of tell us about the role of gonadal hormones and how these influence or may influence clinical measures. She's currently an assistant professor at McGill University. And as an American, I did not take the time to understand the Canadian academic structuring. But anyway, we thank her and I will turn it over to her.
That's great. Thank you for that introduction, Dane. It's okay. The Canadian academic structure is, it's not very exciting. I think it's pretty similar to the US. All right, I'm gonna move you guys over here. So if I look to the side, I'm looking at you guys. Yeah, so thank you so much for the invitation. I'm really happy to be here. I love the idea of a grad student nominated speaker. I'm going to steal this for our seminar. I'm honored. I hope I do you proud. For those, I was told there was a vote, so I hope those that voted for me, I do you proud. So yeah, I'm here to talk about gonadal hormones today on cardiovascular function and health. This title is somewhat of a joke. It's not really a funny joke, except if you're a cardiovascular researcher. I for the last like 10 years or so, I've been going to conferences with going to talks that say, we're going to talk about hormones and cardiovascular function, and be like, that's awesome. I love hormones. I love all of the gonadal hormones. And then the talk ends up inevitably being about estrogen and only estrogen. And now I don't get my hopes up anymore. I assume the talks are going to be about estrogen. And that's not really a chirp, honestly. We know a lot more about what estrogen does to cardiovascular function than any other hormone. But I wanted to really give testosterone the spotlight today. That's why I put commas into my title for the first time. It's the first time I've ever done that. But really focus on testosterone today as it impacts cardiovascular function and health in women. Okay, so I've got a 45-minute plan. I'm going to try to stick to 45 minutes. And I'll try not to talk too fast. My grad students told me I have to cut back on the fun background stories. So I will leave my fun background stories out, and you can ask me for those stories in the question answer period if you want to hear them. So we're going to start with who I am, in case you don't know. And then we'll get into really where the mainstream interest in gonadal hormones came about. It's relatively new that cardiovascular researchers have been interested in gonadal hormones, I would say over the past 20 years or so. So we'll talk a little bit about why that came about, and then we'll get into studying androgen exposure in women and the model that we're using in my lab, which is polycystic ovary syndrome. So this is a condition that is actually quite prevalent in women, and it allows us to study androgen exposure in women. And then we'll talk about the other hormones, and that's another joke because that's estrogen and progesterone. Again, it's not funny. You don't have to laugh. My lab laughed at it because we're all bitter about this hormone thing. But anyway, these are my science jokes for today. Okay, so I wanted to start with a couple of working definitions for today's talk. So first off, gonadal hormones. You might be wondering why I'm saying gonadal hormones. It doesn't really roll off the tongue like sex hormones does. And the reason, and we're talking about androgens, estrogens, and progestogens when we talk about gonadal hormones. I'm not saying it's wrong to say sex hormones. I think, frankly, if you went on my website right now, it would probably say sex hormones on there. The idea, though, is that the term sex hormones sort of insinuates that these are the hormones that dictate sex characteristics. That's not super true. I mean, men have estrogen. In fact, depending on the menstrual cycle phase where you measure women, men have the same amount of estrogen as women. Women have testosterone. Women have more testosterone than they do estrogen. So we know that these are really not necessarily, they don't determine sex. Not to mention transgender individuals, the term sex hormone can start to become exclusive for different populations. So the idea is that gonadal hormones is more of an inclusive term. So just pointing that out. I was just at the Canadian Women's Heart Health Summit, which was all about sort of gender and language that allows us to assess sort of the intersectionality of women's health. So I just wanted to point that out. And then I also wanted to say that I'm going to talk about sex differences a little bit today. When I talk about sex, I'm really talking about the biological attributes associated with physical features, so chromosomes, gene expression, really more of a sex assigned at birth situation. So I might use the term men and women, male, female, but I'm really talking about sex differences. Now, all of these data were collected in humans, and obviously sex is independent of gender. Another thing that if you haven't read into it, I've included a link here, which is from the sort of the Canadian NIH, which is the Canadian Institutes of Health Research. They've got nice statements on the difference between sex and gender. Our research is really interested in gender, sort of masculinity, femininity, how that impacts your cardiovascular outcomes, but something I will not be talking about today. There's a lot that I'm not going to be talking about today. It kind of breaks my heart to have to cut, but I have 45 minutes. So yeah, okay. So those are sort of my disclaimers and working definitions for today. Okay, so let's get into who am I? It's less existential than Zoolander might have you believe. So I'm the director of the Cardiovascular Health and Autonomic Regulation Lab. Our lab was established in January of 2018. I added this picture because I was looking at it for the materials for this talk. And I was looking at it thinking, my God, I've aged like 10 years in the last year of COVID. And I thought, look, she looks so optimistic. She has no idea that this COVID thing is going to hit her, hit all of us, but in year 3 of my lab. So anyway, it was good times before I knew that COVID was coming. So my lab really focuses on the neurovascular control of cardiovascular outcomes. I was trained, like Dave mentioned, in a neurovascular lab with Dr. Kevin Shoemaker. So my really base of understanding is how the neurovascular system controls the cardiovascular system. And then we come at that with our special area of expertise looking at sex differences in hormones. This is my lab from a couple of summers ago, the last time we were allowed to do a photo shoot with all of us in person. We look very happy, again, and naive. Here's my lab as of this past Tuesday. Again, looking very happy. Really tolerating me as I force them to pose for pictures for a talk. So that was a good crew. Okay, so when I'm talking about the neurovascular regulation of blood pressure, this is really what I see in my head and, you know, how we approach the neurovascular regulation of blood pressure. So it's really looking at these efferent sympathetic nerves, which are synapsing with these arterial blood vessels, skeletal muscle arterioles. We call these the resistance vessels. And I know with Dr. Lindbergh in your department, you're probably well aware of this, but just really quick. These vessels are very important for your day-to-day regulation. Right now I'm sitting up, gravity's trying to pull my blood into my lower body. So that's getting detected by my baroreceptors, for instance, which is causing my sympathetic nerve activity to increase and causing those blood vessels to vasoconstrict, therefore enabling blood flow back up to the heart and helping me to not pass out. I'm giving a talk right now. I'm, gesturing wildly. So these kinds of things can be sympathetic stimulants. I know that Missouri is getting some cold weather. Cold stress is a stimulant for the sympathetic nervous system. So your sympathetic nervous system is constantly being activated and deactivated. And so these blood vessels are constantly vasoconstricting and vasodilating. As a general rule, we think of vasodilated vessels as healthy vessels, and vasoconstricted vessels as an adverse response. So high sympathetic activity is generally bad in terms of your cardiovascular health. Vasoconstricted arteries with high resistance are bad in terms of cardiovascular health, and vice versa. I also wanted to point out that when we think about what the sympathetic nervous system is doing, it's really releasing neurotransmitter onto those smooth muscle cells, which surround the arterioles. But don't forget, we have the lumen of the artery through which your blood flows. And that's important when we think about hormones and where and how hormones might exert their effects. And I'll come back to that in a second. Now, I'm presenting some data from our lab today, but as I was mentioning to Dr. Lindberg, I really had grand plans for the last year and data collection, and a lot of it was halted with COVID. So we have some data from our lab here today, also some data from other labs. But what we do in our lab is generally microneurography to measure muscle sympathetic nerve activity. So this is where we stick a small recording electrode into the peroneal nerve in the leg, and we can nestle it right up against those sympathetic neurons and measure their activity. And then we generally use ultrasound to record blood flow. I will be presenting a little bit of ultrasound-derived measures today, but you'll see a lot of MSNA data today. So when we think about neurovascular regulation, we think a lot about sex differences, and even today thinking about the impacts of hormones. And these graphs, this is one of our lab's favorite graphs. These data come from Emma Hart when she was at the Mayo Clinic. Based on the previous slide, this should make a lot of sense to you. So this is correlating someone's muscle sympathetic nerve activity against their total peripheral resistance. So in this example, or in this figure, people who have high sympathetic nerve activity have high peripheral resistance, which makes sense. Individuals who have low sympathetic activity have low peripheral resistance. So these people are not vasoconstricting those arteries very much. Resistance is low. These people are very dilated because they have low MSNA. If you've looked at the top of this graph or you're familiar with it, you know that these are men. And when we look at women, we see something completely different. So there's no relationship between MSNA and total peripheral resistance in young, healthy women, premenopausal women. So these data really suggest that women demonstrate distinct cardiovascular control mechanisms from men. I will tell you, we've put men's and women's hands in ice water, and both of them increase their sympathetic nerve activity. If you have someone stand upright and have that gravity pull blood into your lower body, the sympathetic nervous system activates in both sexes. So the same basic loops are there, but the relationships that we see and the importance of the sympathetic nervous system as a determinant of peripheral resistance and blood pressure seems to be different between the sexes. And if you're wondering why I'm talking about this today, when we look at these data, we're wondering, is this an effect of sex or is this an effect of sex hormones? One of the things that drives me a little bit crazy when people talk about male-female differences is automatically jumping to sex hormones. There are a lot of differences between the sexes that have nothing to do with hormones. And even if you talk about, men having a lot of testosterone and women less, or men and women having the same levels of estrogen, if you measure women in the early follicular phase of the menstrual cycle, we don't know if they have the same number of estrogen receptors, if they have the same activity of estrogen receptors. So, even normalizing men and women to their concentrations of hormones doesn't really allow you to isolate the effect of hormones. So my quick disclaimer that any effect of sex is not just hormones, but there's pretty good evidence that what we're seeing in women could be hormones at work. And in terms of sites of activation, I've made some arrows here. Some of this was last minute research yesterday and today as I was kind of looking at where we have evidence for actions of hormones. We know that, and honestly, most of this is estrogen, I'm going to tell you. But we know one of the most well-established impacts of estrogen is as a vasodilator. So if you were to pick up any review paper and read about the vascular consequences of estrogen, it would say estrogen is a potent vasodilator. It upregulates endothelial nitric oxide synthase, which means that it's promoting vasodilation inside this artery. And if you think about the sympathetic nervous system trying to vasoconstrict from the outside, it just means that the estrogen is sort of opposing that and making it harder for the sympathetic nervous system to constrict, or requiring a greater amount of sympathetic activity to constrict. Hormones also are found in the brain and brainstem. So if you notice the brain sitting over here, we know that the brainstem is the final site of regulation for sympathetic outflow, or at least preganglionic sympathetic outflow. We know that there are estrogen receptors all through the brainstem. So there's a potential site of regulation of sympathetic activity. People like Kevin Shoemaker, von Masefield have really shown the cortical integration sites for sympathetic activity in humans. And we know that there are estrogen receptors there. If you're familiar with any of the insular cortex work, there's data showing that if you inject estrogen onto the insular cortex, you get a sympathetic response. So we know that there are estrogen receptors everywhere. My last minute Googling told me that there are estrogen receptors in the ganglia. And we know from Mayo Clinic, again, work that basically with menopause, you have a change in the densities and the distribution or distribution. We don't really know which, if it's the sensitivity, the number of them or the sensitivity of them. But we have a change in receptor balance pre and post menopause. And anything that happens around menopause sort of suggests an effect of hormones. So hormones might affect your adrenergic receptors, which are the receptors that your neurotransmitters are going to bind to once they're released from these sympathetic neurons. So essentially, what I'm trying to say is every single part of this diagram has the potential to be impacted by hormones. And again, when I say hormones, most of this evidence is estrogen. So really, you're seeing that my joke from the first slide is kind of a terrible joke, because we know a lot about estrogen and very little about progesterone and testosterone, but I try anyway. Okay, so within my lab, we have two central hypotheses having to do with hormones. So the first is that gonadal hormones directly modulate neurovascular function. And the second is that sustained or chronic changes to this hormone milieu are going to result in clinically significant changes in cardiovascular health. So that means in populations who have sort of sustained or chronic changes to the hormones, that we're gonna start to see outcomes, changes in cardiovascular disease risk, changes in cardiovascular morbidity and mortality. So our lay hypotheses are, and this is very lay, are in women, estrogen is good, testosterone is bad, and progesterone is ... Yeah. Really, I think this is about where we're at in terms of progesterone and what progesterone does. Every year I get a student to write a paper on what are progesterone's effects on the vascular system and cardiovascular system, and they always kind of come up with, I don't know. All right. So that was a little bit of who I am and what my lab does. I guess it was more about my lab. So now let's hop into the interest in gonadal hormones mainstream, and why is my lab studying these gonadal hormones? I'm a little bit of a comic book and especially comic book movie nerd. So I like the idea of an origin story a la comic book superhero. So this is really where the comic book references are going to end. But going back to the origin, to me at least, of the cardiovascular field's interest in hormones. Basically, we've known for quite some time that cardiovascular diseases are less common in premenopausal women than in men. It's one of my favorite figures showing the incidence of hypertension across the decades of life. You can see this is for hypertension, obviously, but you can find very similar figures for a number of cardiovascular disease states. And you can see that men who are in the white bars here have much higher rates of hypertension than women at these different ages. However, at the age of menopause, we start to see women catch up to men in terms of their cardiovascular disease rates, in this case hypertension, or even surpass them, which you can see in this over 70 group. So when we start to ask why this is the case, a lot of people have pointed, I think, understandably, to estrogen. So here's a nice figure I found the other day showing what happens to estradiol over the lifespan. And sort of this increase that occurs shortly after puberty, and then this decrease that happens at menopause, which I think is pretty common knowledge at this point. So this idea that when you start producing less estrogen, you lose this cardio protection. So premenopausal women are afforded this cardio protection relative to men. It's mediated by estrogen. They go through menopause, they lose their estrogen, they lose their cardio protection. It's a very neat idea. And then the idea becomes, all right, well, if we want to prevent cardiovascular disease in that population, let's just give them hormone replacement therapy and everything will be great. So hormone replacement therapy was given, quite ubiquitously through the 90s. Hormone replacement therapy is estrogen and a progestin. It's generally, there's only a couple situations where you can really give what we call unopposed estrogen, so estrogen on its own. In the 90s, we thought, I was 6 in 1990, but the field thought, let's do the large-scale randomized clinical control trials to figure out what hormone replacement therapy does in terms of cardiovascular health. So this includes the heart and estrogen replacement study and the Women's Health Initiative study. And yes, I'm looking at my notes to remember those acronyms. And what they found was actually that combined hormone replacement therapy, so estrogen and progesterone, and progestin, were not effective in preventing cardiovascular disease. And even more than that, they increased the incidence of some cardiovascular diseases. This was huge news. Women all over the world were taken off of their hormone replacement therapy. But then people were reading these papers and correctly pointing out that there were a number of methodological issues with these studies. And so follow-up studies like the Chronos Early Estrogen Prevention Study, which looked at women with no cardiovascular risk factors, and the Elite studies, this is early versus late intervention trial with estradiol, they wanted to address the limitations of the previous trials, including the Women's Health Initiative study. And basically, these studies found that the effects of hormone replacement therapy depend on the timing, the dose, the formulation, the presence of pre-existing risk factors. So essentially, a lot of things. Are you initiating hormone replacement therapy immediately after menopause? Are you waiting 10 years? What's the dosing? What are you putting into this? I would say that the subtext and really the take-home message, if you've fallen asleep through this slide, is that the cardiovascular actions of hormones are complex and incompletely understood. Really, I think the hormone replacement therapy trial, and I've talked to clinicians who were on these initial papers, that they sort of surprised us when these results came back. And the real take-home message was we have a lot more to learn about these hormones. And honestly, that was true in the 2000s, mid to late 2000s, and I would say it's still true today in 2021. So switching gears slightly to my origin story, my interest in this, I, in the mid to late 2000s, was a master's student in Kevin Shoemaker's lab. There's Charlotte, 2009 Charlotte, standing with my science hero, my father. We're feeding fish at the pond, if you're wondering what I'm holding in my hand. I'm really displaying that proudly. That's the only picture I could find of myself in 2009. So in 2009, I'm a master's student, and I'm starting to get interested in, basically, I'm reading all of these Women's Health Initiative studies, and I'm going like, I cannot believe that we don't understand hormone replacement. And I had a mother who was postmenopausal at the time. Should we be giving her hormone replacement therapy? So I'm reading about this, and this conference comes up. It was an APS conference in Colorado, targeting sex, steroids, and gender in cardiovascular physiology. So I went to this conference, and this is one of my regrets in life. I really wish I could remember who gave the statement. I know that Jane Reckelhoff was there, but I don't want to, falsely attribute this. But I remember a speaker saying, we always think about menopause as a time of estrogen depletion, but we need to keep in mind that what menopause is a change in the estrogen-androgen balance. So this is actually how I found this figure. I was trying to find a figure that showed us both estradiol and testosterone. You can see this very nicely, that testosterone increases in women at puberty and then decreases, but it decreases less abruptly than estrogen does. And the result, the reason for that is, or one of the reasons for that is you can produce testosterone at the adrenal glands. So you start to become more androgen dominant than you were as a premenopausal person. So the speaker was saying you really have to think about the postmenopausal period as a change in your estrogen-androgen balance. My mind was blown. I was like, I've never even thought about testosterone in women before. This is fascinating. Tell me more. So I started thinking about postmenopausal women, and The problem with using the postmenopausal period to study androgens is, I think, very well exemplified by this paper that came out last summer with Kevin Shoemaker's on there and Dan Keir, which really showed the change in sympathetic activity that happens across the lifespan in men and women. So in this study, they went back and did a retrospective analysis on data from multiple labs, starting from 1986 all the way up to present day. And what they found is that the pattern of changes in sympathetic nerve activity looked different between men and women. So I actually really like the summary slide on the left. On top, the women are in the red, and it shows this dip in sympathetic nerve activity that happens in young women that starts to go up and then increases over the lifespan. Whereas in men, they don't have this early dip and sort of a slower rate of increase. And this mean difference between the sexes showing that actually sympathetic activity can be higher with postmenopausal women than similarly aged men. Here's the raw data with all the data points over here. But one of the things that I actually found myself on a plane next to Dan Keir when he was analyzing these data, and we were talking about menopause and hormones, and he said, you know, what's really interesting when you look at these data is that the increases in sympathetic activity that we see in women, they precede menopause. So you see them start to go up. You know, the average age of menopause is 53. It's already pretty high at 53. So there seems to be something to do with aging that increases MSNA. And so really, it's difficult to tease apart the impacts of aging and androgen-estrogen balance in postmenopausal women. Before I move on from the slide, I just want to point out one thing, and this is going to sound like I'm ripping on these guys. I'm really not. I just want to point out the number of dots in the men and the women figures and how many fewer dots there are in the figure of women. One of the things that really alarms me as a 36-year-old person is the gap right in here in this 40 to 50 range. This sort of middle age, dare I say, age range, we seem to know very little about what's going on in terms of sympathetic activity. I'm not saying no one has data here, but you can see in general, there's been a lot less research studying women than men. And so this is one thing that I would like to point out, that we still need more basic research on sex differences and what's going on in women from a neurovascular perspective. Okay, so this issue with using menopause as a tool, let's say, to study androgen-estrogen balance, led me down a PCOS rabbit hole that, you know, whatever it is, years later, 12 years later now, I'm bad at mental math, guys, I still have not come out of. I'm still really interested in polycystic ovary syndrome. Okay, maybe this is a good time for a drink of water. So PCOS is the most common reproductive endocrinopathy in young women, affecting 6 to 20% of women. I know that's a very big range. The stat you'll usually see is 10%. You'll see in a second why there's this big range of diagnostic criteria. And that is because, first of all, I haven't gone over all the different diagnostic criteria. The Rotterdam are the most common, so there are other criteria to diagnose PCOS, and that adds some of the variability that you see in the numbers. Also, the Rotterdam criteria only require 2 of 3 criteria. So we've got criteria about what the ovaries look like, whether someone's ovulating, and then hyperandrogenism. So clinical is having the clinical signs, so excess body hair, hair loss, that kind of thing. Biochemicals, just a blood draw, and elevated levels of testosterone. So this is obviously very relevant to today's talk, and that 75% of women-ish with PCOS are hyperandrogenic. This is a very common aspect of PCOS. Also important to today's talk is that PCOS is associated with lots of cardiovascular risk factors. So obesity, insulin resistance, arterial stiffness, and endothelial dysfunction. I'm laughing at insulin resistance because I didn't put it in, and I know. Anyway, we'll talk about it. We'll talk about it in the questions. Okay. So arterial stiffness, endothelial dysfunction. We know that there are these potent risk factors that are pretty common in women with PCOS. So for instance, if we look at the effect of PCOS on endothelial function, this is a meta-analysis that was done in 2013, back before meta-analyses became all the rage in COVID 2020. I don't know if anyone out there is doing one. We're doing one in our lab right now. Everyone's doing a meta-analysis right now. Anyway, so this was a meta-analysis on studies that had done flow-mediated dilation studies. So this is just very quickly a test of endothelial function. You use ultrasound, usually on the brachial artery, not always. And basically you elicit a hyperemic stress by inflating a blood pressure cuff, including the limb for 5 minutes. Then you release it, get this huge influx of blood flow, and that causes the brachial artery to reactively vasodilate. And then you measure the magnitude of that dilation, and that tells you, like, an artery that dilates a lot is a very healthy artery. An artery that doesn't dilate a lot is not a healthy artery. And there's lots of studies that show, actually, like I say down here, that the risk of a cardiovascular event, so this is not a PCOS statistic, this is just a general statistic, the risk of a cardiovascular event increases by 21% for every 3.5% decrease in FMD, which is pretty mind-blowing. So these authors did a meta-analysis of studies in PCOS, and these are studies that only use the Rotterdam criteria for diagnosis and that matched the PCOS women with the control women for age and BMI. So they tried to be as rigorous as they could with the studies. And, especially considering that women with PCOS tend to be obese, you need to make sure that you're sort of accounting for the impact of obesity there. And with those sort of strict measures, they found approximately a 4% decrease in FMD in the PCOS women relative to controls. So this shows that endothelial function is impaired in women with PCOS, even when matched for BMI with the controls. So another interesting part of PCOS is that it's associated with an increased cardiometabolic risk. And I know I said early on in the presentation that there are issues with considering postmenopausal women a good model for studying estrogen-androgen balance. I found out not too long into my PCOS journey that PCOS isn't really a great tool to study androgen specifically either, because there's a lot of other stuff going on. But now I'm hooked and it's too late. So basically, PCOS is also associated with a cardiometabolic risk, and that is heterogeneous, like the heterogeneity of PCOS. So to explain what I mean by that, here's a really nice Venn diagram showing the 3 different diagnostic criteria. So the hyperandrogenism, the polycystic ovaries, and the ovulation, irregular ovulation. And obviously the different phenotype or the different types of PCOS that you can have based on those criteria, which is to say there is a type of PCOS that is not hyperandrogenic. So this is, if you have these 2 criteria, the polycystic ovaries and the illegal ovulation, you have PCOS. Now, we know that the severity of the consequences of PCOS are tied to the phenotype. And in fact, those associations going from weak to strong are the weakest in the phenotypes that are not associated with high androgen levels, which is all to say that the most severe consequences of PCOS appear to be associated with hyperandrogenism. So as much as there are cardiometabolic issues, including insulin resistance and obesity, which are obviously tied to one another, we know that the hyperandrogenism seems to play a potent role in mediating that cardiometabolic risk. I'll just pause here just so I don't forget to say it. If I hook you on PCOS and you really want to read more, this is my favorite PCOS review article. It's beautiful. I come back to it all the time. You're going to see more diagrams from that article, like right now. So in this review article, Eskimo Morale presents this hypothesis that we might be, when we're talking about the different phenotypes, lumping together a number of conditions under one umbrella, and that we might be talking sort of about 2 different types of women with PCOS. So on one hand, we've got this woman in the blue, and she has a PCOS disorder that's so severe that even in the absence of triggering factors like abdominal adiposity, insulin resistance, and obesity, she presents with a PCOS phenotype and with the consequences of PCOS. Whereas at the other end, here in the red, we have a woman with a mild defect in androgen secretion, but that defect gets amplified by triggering factors. So if this woman were to be lean and she would go into the doctor's office, she would not be diagnosed with PCOS. She would not meet the criteria for PCOS. But when she gains weight, has an abdominal adiposity, becomes insulin resistant, becomes obese, that's when this PCOS shows up. So this is going to be important as I show you the next studies that are coming up here. So we might be dealing with two different things, and those two different sort of severities of PCOS might be related to the hyperandrogenism and problems with androgen secretion per se. So in my second postdoc with Dr. Nina Stackenfeld, we looked at ethinyl estradiol supplementation in hyperandrogenic women with PCOS. So we specifically sought out hyperandrogenic women with PCOS, and we tested two groups, lean women and obese women with PCOS. We then supplemented women with ethinyl estradiol for 7 days. We did this because many women with PCOS are treated with hormonal contraceptives. I would say those are one of the most common treatments for PCOS. And we can talk about why women with PCOS are prescribed hormonal contraceptives. But ethinyl estradiol is the estrogen component of those contraceptives. Remember, estrogen is vasodilatory. So we thought, well, let's look at their FMD. Surely ethinyl estradiol will improve endothelial function in these hyperandrogenic women with PCOS. That's what estrogen does. And I should pause and say these data actually, much to Nina's chagrin, I have not yet published, but it turned out to be good because one of my master's students who was going to look at PCOS as her master's project, when COVID hit, ended up in trouble without data to analyze. And I said, Nina, why don't we get her to analyze these data? So my master's student, Danielle, has really been a hero analyzing these data for us. So there you go. My message to the grad students is don't let your supervisor make you feel bad for not analyzing data. I'm just kidding. It's really bad. And I was a postdoc. I really should have. All right. So we have these data in obese women, obese controls on the left and PCOS on the right. So just look at these black circles first. You can see, I mean, the numbers are small here, but you can see that the PCOS have, let's say, the same FMD as the control women. There was no main effect of PCOS here. And in both cases, when we give obese women ethinyl estradiol for 7 days and we reassess their FMD, there's an improvement in FMD. That was a main effect of estrogen. And you can see a pretty consistent response here where basically everybody improves their FMD. So clean. So then we look at lean women. So first of all, please ignore the open squares for a hot minute. We'll look at the controls here. So here are the FMDs in our control women. So lean controls. And here are our PCOS, our lean PCOS. And you can see, whereas there was no effective PCOS in the obese women, they had the same endothelial function. Our lean women had a significantly reduced FMD compared to the controls. This was an overall effective group. So this is really telling us that is endothelial function worse in lean women with PCOS than it is in obese PCOS? And so I wouldn't even say, that their baseline levels of estrogen, of FMD, are that much worse. There's some variability there. But the real problem is that they do not respond to estrogen. So some of the women increase, some of them decrease. So there's really no main effect of estrogen. So whereas estrogen was able to improve endothelial function in the obese women, it did not do anything in the lean women with PCOS. I would love to move on and not talk about that asterisk sitting there, but that is, yes, a negative effect of ethinyl estradiol on FMD in the control women. I would love to talk about it in the question and answer period if you want to ask me about it. Right now, I'll just say hormones are crazy and we really don't know a lot about hormones. Anyway, if you're curious about that, let's chat about it. But for now, I'll move on because we're talking about PCOS here, not the effect of hormones and controls. Okay, so sorry, I should just really quickly say, the take-home message of the slide is that endothelial function, we have some evidence that it's worse in lean hyperandrogenic women with PCOS compared to obese hyperandrogenic women with PCOS. And anyone who's researched obesity, I mean, they can tell you we never see that. You know, obesity is in and of itself a cardiovascular risk factor. Obesity in and of itself is usually associated with, things like endothelial dysfunction, increased MSNA. So it's very surprising that we're seeing results like this. This is not our data. This is one of my favorite papers that really got me interested in PCOS. So this is a study from 2008, actually looking at lean women with PCOS only. There's no overweight or obese women in the study. And they measured muscle sympathetic nerve activity, and they found that women with PCOS had higher sympathetic activity than the controls. I pressed the wrong button, sorry. And that this was correlated to their testosterone levels. So the women with PCOS are in black, the controls are in gray, and you can see actually a pretty nice relationship here between testosterone and their sympathetic nerve activity. So testosterone was predicting the level of sympathetic nerve activity, again suggesting that hyperandrogenism is directly mediating an impact on the neurovascular system. So our lab, we've just been getting started with this, like I said. We're really interested in lean PCOS for all of these reasons. We think that we're looking at the most severe phenotype of PCOS. We also think it's a nice way to sort of isolate the impact of androgens to the extent possible. Many lean women with PCOS are still insulin resistant, so it's not a perfectly clean model of elevated androgens. But it's nice. It lets us look at PCOS without any kind of confounding impacts of obesity. And again, we think it is the more severe PCOS phenotype. So we've been studying them and looking at cardiovascular reactivity. So, like I mentioned, I'm sitting at my desk right now, my sympathetic nervous system, I'm sure, is firing like crazy. Your sympathetic nervous system is very seldom quiet. And so, we think that looking at how the sympathetic nervous system excites is a very good measure of cardiovascular health. And there's lots of data to support that if you measure someone's reactivity to stress, it will predict their cardiovascular health later on. So here's one of the stressors that we do. This is an apnea. So it's just a voluntary breath hold as long as they can hold it. As you can see this, if you're familiar with sympathetic nerve tracings, it elicits a huge increase in sympathetic activity. It's one of the reasons we like to do it, sort of near max. We usually divide this up into two halves, an initial and a latter half. You see a pretty consistent biphasic response where the biggest responses are at the end, right before you can't hold your breath anymore. And when we did this in lean women with PCOS and lean controls, what we found is that at the beginning of the apnea, it wasn't significantly different between the PCOS and the controls. Now, I think this is like 4 or 5 people in each group. So these are preliminary data. So don't get too excited. This is a conference publication down here. We are obviously going to increase our sample size, but what you see pretty convincingly, I think, is in the latter half of that apnea, a huge increase in sympathetic activity that we didn't see in the control women. So these lean hyperandrogenic women with PCOS demonstrate these markedly larger increases in sympathetic activity during this severe phase of the apnea stress, chemoreflex stress. And honestly, even more worrying than that to me is the fact that blood pressure, so this is diastolic blood pressure, was higher in the PCOS than the controls at both time points. So, we talked about the transduction of a sympathetic signal into a vascular outcome like total peripheral resistance. We said that if sympathetic nerve activity increases to increase resistance and increase blood pressure, but these women with PCOS have higher blood pressure even when their sympathetic nerve activity is not that much higher than the controls or not significantly different than the controls. So even in the absence of difference in MSNA, we have higher diastolic blood pressures, and this is suggesting really exacerbated neurovascular transduction in the lean women with PCOS relative to controls. That first slide I showed you with Emma Hart's data, this is sort of taken to really demonstrate the cardioprotection that women have. It doesn't matter what their MSNA is, they keep a very constant peripheral resistance. So they're sort of protected from high MSNA in that way. It seems like these very preliminary data seem to show that these women with PCOS are not afforded that same protection that healthy premenopausal women without PCOS are afforded. So to summarize all that PCOS data, there's good evidence to say that PCOS confers an increased cardiovascular risk and that androgens are responsible for at least some of this. I would argue more than some. When we control for obesity or only test lean women with PCOS, we see impaired endothelial function, an endothelial function that actually might be worse in lean women with PCOS than obese. That muscle sympathetic nerve activity is elevated at rest and in response to severe chemoreflex stress. and that's paired with high blood pressure. So I just put these figures in here. My other slide was full, showing that, we don't have the data set to run these regressions yet, but our, low-key hypothesis is that our young women with PCOS are going to look more like young men when you plot their neurovascular transduction than they are like normal premenopausal women. So yeah, that's sort of where we're going with this. Okay, so I'm running out of time, which is perfect time to bring up the other hormones. I'm going to go over these in a lot less detail. I feel like I'm sticking it to estrogen and progesterone, and I mean, they're hormones. This is, anyway, I really love the idea of giving the spotlight to testosterone. It's doing my heart some good, I have to tell you. Okay, so the way that we've been studying hormones, or that I've been studying hormones to date in my career, is using the menstrual cycle. And the menstrual cycle is pretty convenient because it involves cycling of estrogen and progesterone. So the menstrual cycle phases that we like to look at are the early follicular phase, when estrogen and progesterone are low. So estrogens are green line and progesterones are maroon line, versus the mid-luteal phase here, where both hormones are high. So it gets you can kind of compare a low hormone versus a high hormone phase. So these are data that we published, that I published as part of Kevin Shoemaker's lab. This has been shown by lots of people at this point, that mid-luteal MSNA, so when hormones are high, is higher than early follicular at rest. So not all studies have found this. Some studies have found similar MSNA between the phases, but no one's ever shown greater resting MSNA in the early follicular phase. So it seems that something to do with the mid-luteal phase, maybe the high hormones, but that's associated with an increase in sympathetic nerve activity. Interestingly, it's not associated with any hemodynamic outcomes. So total peripheral resistance, for instance, is the same between menstrual cycles, which suggests that neurovascular transduction is blunted in that mid-luteal phase of the menstrual cycle. So when hormones are high, transduction is lower. So something about hormones are reducing neurovascular transduction there. So when we ask why the menstrual cycle might affect MSNA, this is, I know I've said this a couple times now, this is one of my favorite papers, but this is one of my favorite papers of all time. In this paper, I just think this is a bananas study design. I'm dying to replicate it, but it would be so difficult to do. I understand why no one's really done this. So they did a hormone suppression. So they used a GnRH antagonist, so a gonadotropin releasing hormone antagonist, which is just to say they blocked hormone production. So they measured women 3 times in their mid-luteal phase, so when their hormones were high. Then they blocked their hormones for 5 days, measured them again, measured their MSNA again. Then they added back either estrogen through a transdermal patch or progesterone orally. And what you can see is that, so after the gonadotropin, so the gonadotropin antagonist decreased MSNA from the mid-luteal phase, again, implying that something about these hormones, estrogen and or progesterone, had been increasing MSNA. And then when you block their production, MSNA comes down. Not in everybody. Hormones are crazy. And then, so the estrogen is in this dashed line. You can see, fairly convincing, I would say, that estrogen decreases when you add back estrogen. So it seems like estrogen is decreasing MSNA. The progesterone, now we have 1 person going down, 2 people going up. These data kind of suggest that estrogen reduces MSNA and progesterone increases MSNA. And now, these are very small sample sizes. These are very difficult data to get. I will admit to more data that I haven't published. This is a terrible talk. I'm a terrible example. Sorry. So these are data from my PhD, where we, those same data where we looked at resting MSNA. So we correlated the change in progesterone across the menstrual cycle against the change in MSNA across the menstrual cycle. And I mean, this is a fairly small number of individuals, but what we found is that the magnitude of change in progesterone predicted the change in MSNA. So people who had a really big increase in progesterone from the early follicular to the mid-luteal phase had a big increase in MSNA across the menstrual cycle, and vice versa. So we had, the 1 person who's decreasing MSNA across the menstrual cycle, or 2 people, I should say, those people had very small increases in progesterone. So does progesterone increase MSNA? I don't know. ... So why would the menstrual cycle affect neurovascular transduction? Well, we know again that estrogen has very well-established vasodilatory effects. One of the things that I would say here is actually, I've worded this vasodilatory effects of estrogen active buffer increased MSNA. It's, I think, likely that estrogen is vasodilating the arteries and MSNA is increasing to compensate and keep that TPR constant. And we saw that TPR was the same across menstrual cycle phases. Like I've said, there is data from Mayo Clinic, these are Heart data, that beta adrenergic receptors affected by hormones. I mentioned those briefly. We know that beta adrenergic activity or density is affected by menopause. So I don't know, honestly, if beta receptors change across the menstrual cycle. I couldn't tell you. I don't think anyone could tell you, but I could be wrong there. I don't think anyone knows. Okay. So now what if we expose women to stress across the menstrual cycle? So let's use the same apnea stress. It's actually not exactly the same. This was data from my PhD where we really wanted to drive the sympathetic nervous system as hard as we could. So instead of just getting them to hold their breath, we had them rebreathe in and out of rebreathing bags so that they became hypoxic, hypercapnic. Then we had them hold their breath. So we were really trying to drive that sympathetic nerve activity as high as we could. And again, we divided the apnea up into two halves. And what we saw was huge increases in MSNA, as per usual. But you can see this is a representative tracing of this individual who is firing like crazy in these huge bursts in this early follicular phase. So if I show you the group data, we actually found that the MSNA response to this apnea, this acute chemoreflex apnea stress, was greater in the early follicular phase than the mid-luteal phase. And this is a main effect of hormone, by the way. Here are the hemodynamics. They're the same between menstrual cycle phases. So again, nothing happening in terms of expression as TPR. But these suggest that the sympathetic responses are exaggerated during the low hormone phase and not reflected as an increase in TPR blood pressure. But remember, I just told you that baseline MSNA was greater when hormones are high in the mid-luteal phase. So this implies that menstrual cycle-induced changes in MSNA are not unidirectional. Or to put it another way, if we're saying that hormones are responsible for the effects on MSNA that we're seeing across the menstrual cycle, it is not a blanket response where high hormones mean high MSNA and high reactivity. There is something going on where they're potentiating the response to a chemoreflex stress when there's low hormones, or high hormones are restricting the response to a chemoreflex stress, I don't know. And that at rest, they're elevating MSNA. So if I've lost you at this point, welcome to the club. One of my favorite Star Wars memes. Hormones are crazy. Okay, so I love ending my presentations on a note of confusion. What did we learn about gonadal hormones and cardiovascular outcomes? Well, I would say estrogen is good. Estradiol is good most of the time. We're still looking at people are still looking at hormone replacement therapy. Again, unopposed estrogen, it has to be very situation specific. But in general, I think that estrogen is good for the cardiovascular system. We generally think hyperandrogenism is bad. Again, why do we see it in these lean women and not these obese women with PCOS? I don't know. But yeah. Progesterone, we continue to ... I have a good Virginia Miller story, which is at a conference, there was a talk, I think it was Jill Barnes who was moderating the discussion after, and I got up and asked a question about progesterone. I said, you've talked a lot about estrogen. What do you think progesterone is doing here? And she leaned over and said, I'm going to turn this over to Virginia Miller. And if anyone who knows Virginia Miller knows she is the godmother of hormones. One of my all-time science heroes. And she leaned into the mic and said, this field is wide open. I have no idea, and I would encourage you to research this. And I thought, okay, Virginia, I'll do it. So yeah, we don't really know a lot about progesterone. So we still have a lot to learn. But something we do know, so we don't end on unknowns, is that postmenopausal women and women with polycystic ovary syndrome are at increased risk for cardiovascular disease. Cardiovascular disease is still the largest source of morbidity mortality in women worldwide. This is in Canada and the US. It's underappreciated many women don't recognize their risk of CVD. Half of women, this reference I have here shows that half of women don't know that cardiovascular disease is the leading cause of death in their sex. Women are less likely to be diagnosed with cardiovascular disease. There are horrible studies that show that when women go into the hospital having a heart attack, they take longer to be diagnosed with having a heart attack. They're less likely to be believed by their physicians. They're less likely to be treated once diagnosed, so they're less likely after a cardiac event to receive aspirin, ACE inhibitors, and statins. And as a result, they have higher rates of mortality relative to men. So for instance, for coronary heart disease, this is true. And this has been, primarily because women have been underrepresented in large clinical trials over the years. And most of the research has been done on men and extrapolated for women. And this lovely woman on the right is my mother. She is a postmenopausal woman. She's a singular woman, and she and everyone who's postmenopausal right now is having to figure out whether they should be on hormone replacement therapy. And if you're not thinking about postmenopausal women, think about me. I'm going to be 40 soon. What are we doing? What should we be doing with my hormones? And what about in 15 years when I hit menopause? So this is really to tell the trainees out there that I know we've been hearing a lot about women's health. I would still consider this field sort of wide open. There's a lot to learn here in terms of hormones, and it's a really interesting field if you're interested in getting involved. So with that, I'll wrap up and acknowledge my funding sources. We couldn't do any of this without funding, something you really realize once you become a PI and you have to write grants all the time. These were, my graduate, postdoc, and now my lab funding sources. My training mentors, I'd obviously be nothing without them. Kevin Shoemaker taught me everything I know about neurovascular function. And Dr. Stan Van Oom, who was on my advisory committee, my PhD, and was the only one among all of us who knew anything about hormones. So Stan did some good work there. My first postdoc supervisors, Dr. Craig Steinbach and Dr. Margie Davenport. I'm really sorry if anyone came today thinking I was going to talk about pregnancy. They've got great data going on. I really recommend you check them out. And my second postdoc supervisor, Dr. Nina Stackenfeld, who has always been one of my science heroes, and Dr. Jeff Bender and Hugh Taylor, who are profs at Yale, who have been huge in terms of understanding the vascular consequences of hormones. And of course, my lab, past and present, lesson to all the trainees out there. When you get your own lab, take formal lab pictures, because otherwise you've got a bunch of pictures of you guys drinking beers and on Zoom. Anyway, so that is all I have. So thank you. I wasn't quite 45 minutes, but that's it.
Awesome. Well, thank you so much. If everyone could unmute and clap for a moment. Awesome.
Thank you.
Clap emoji works too. So our chat box is full of questions. But first and foremost, thank you for that excellent presentation. We'll just go ahead and jump right into it. Dr. Padilla, this is a spicy question. Is sympathetic vascular transduction different between males and females?
Is sympathetic vascular transduction different? So you mean like beat by beat, or yeah?
That's what the figure that you display comparing side by side MSNA and TPR prompt me that question of whether, if you do the analysis of transduction, would you see, if you follow a burst of SNA, do you see this being followed by a greater vasoconstriction in males versus females? Has this been done? Maybe Paul Fadel has done that, but
yeah. It's been done by a number of people. I think that as it's coming out, it's looking, I'm really going right to the in-between burst thing, because that's what we were recently looking at. But I think that a lot of the responses are similar in terms of the peak blood pressure and vascular responses that you see in men and women. Our recent data are showing that where we start to see sex differences is in between bursts. So when there aren't bursts of activity, that men's blood pressure falls very quickly, whereas women seems to stay a little more stable. And even after a burst, we see that women their blood pressure stays higher a little bit longer than the men. So we're seeing sort of these more volatile patterns of blood pressure reactivity following bursts of sympathetic activity in men, which we're hypothesizing might have to do with why men have higher baseline sympathetic nerve activity. Their vascular responses are a little more volatile following it.
Would you say that maybe women can support the blood pressure via changes in cardiac output, more so that by changes in SNA.
Yeah.
They have to be able to maintain that blood pressure.
Yeah, exactly. Yeah, so one of the very interesting things, I don't know if this is my bias from having been in a pregnancy lab, is that women are sort of built with the ability to be able to maintain fetal blood flow. And so if you think about regulating your blood pressure and supporting your blood pressure from a peripheral standpoint, giving your peripheral blood vessels sort of that ability to constrict and dilate, to regulate blood flow, becomes dangerous when you've got a fetus that's demanding a lot of blood flow. So the idea is that women might regulate more at the level of the heart, so, as it's sort of like a safer system.
That's great. Thank you for sharing.
No problem.
Nice presentation.
Thank you.
Yeah, and to follow that up with another Dr. Padilla question, he asked, does systemic infusion of testosterone increase sympathetic nerve activity, specifically in young healthy females? Or has that even been done?
This is a great question. I don't think, I know that if you, so Nina Stackenfeld, for instance, is getting into researching transgender men. I stayed away from that for this presentation for a couple of reasons. I actually got very interested in what would happen, what the cardiovascular profile of transgender men looks like. It looks like chronic infusion of testosterone does increase or decrease FMD and increase cardiovascular disease risk. Now, I've talked to some clinicians about this, and they would caution, and this is sort of part of the reason I didn't really put it in, because I thought, it's tricky not being an expert in transgender health, but basically it seems to really matter whether an individual has gone through puberty before you start this transgender treatment. So someone whose sex assigned at birth is female, who goes through puberty, who then you start with testosterone treatment, that looks different from a cardiovascular consequences perspective than someone who, if you blocked puberty at the onset and then started administering testosterone, years later. So the short answer is yes, people looked at chronic infusion. I'm not sure about short-term infusion, and it does seem to negatively impact cardiovascular health, but there seem to be ways of getting around this in terms of the timing of hormones.
Thank you very much.
You're welcome. I think the idea of short-term versus long-term hormone exposure is something I'm still wrapping my head around. And the menstrual cycle, and we're looking at FMDs across the menstrual cycle. To what extent would I expect an acute change in estrogen to affect blood flow versus some, chronically looking at levels of estrogen and what chronic estrogen exposure does is something I think a lot about. So these are great questions.
Yeah, and you inadvertently answered another question as well, but we'll come back to that in a moment. So from Dr. Kevin Cummings, do you think that understanding this dip in muscle sympathetic nerve activity in females from age 20 to 30 may be just as important as the gradual rise in females with aging? Could it be due to something with pregnancy, so that specific decade between 20 and 30?
Yes. Yes. I mean, I think we should understand all of these things. I'm kind of biased. The pregnancy thing is a great question. I will tell you with 100% certainty that none of the people in that study were pregnant at the time. Now, what pregnancy does to your sympathetic nerve activity over time, so those are your childbearing years. When I look at that graph, I think, number one, what is happening in those 20-year-olds? And I think all of us who have looked at sex differences in MSNA and not found it in young healthy people can breathe a sigh of relief that we're not the only ones. There seems to be a lot of variability in those young people. When you get into that age group, not to give you all of my grand ideas, but I think that what starts to happen, sort of in that like 30, 40, 50 range when you're premenopausal, but so I guarantee you some of the people in those figures have PCOS and didn't know. Some of them were probably pregnant and in the postpartum period. And we know that if you've had a preeclamptic pregnancy, you're predisposed to early onset cardiovascular disease. We've done some work in there showing changes in sympathetic reactivity in women who have been formerly preeclamptic, and but it's not reflected in blood flow. So there might be sort of these like sort of preemptive changes in sympathetic nerve activity that get sort of unveiled later as vessels start to stiffen. Yeah, so I think it's actually what I'm really impressed about with that dip in MSNA is that I think there's so much variability in those young women and in the life experience, and that it really is where we're lacking a lot of data, I'll also point out. So I'm surprised that they were able to see that dip as clearly as they were. And yeah, 100%. I think whenever I think about sex differences in neurovascular regulation, I think if we can understand how women are cardioprotected, it might give us some clues about how to protect men from cardiovascular disease, since they get a lot more cardiovascular disease at those young ages.
Awesome, thank you. So the next question is from Dr. Lindberg. And so since you've done this, her question is, what are your thoughts on studying across the menstrual cycle as a way to explore the cardiovascular effects of the gonadal hormones? And she's thinking that can we actually isolate these hormones, or is what we're seeing per se in the late follicular phase or early follicular phase a remnant effect of something that just preceded it, basically?
Yeah.
Carryover effect.
Another thing I think about all the time. Yeah. Yeah, I think about sex hormones a lot, or gonadal hormones a lot. I think so. One of the things where we always want to look at is the late follicular phase of the menstrual cycle. I think if anyone here has tried looking at the menstrual cycle, you'll know that it is very difficult to do. Predicting menstrual cycle phases and tracking them. We actually recently did a study where we tried to hit women in the test women in the early follicular, late follicular, and mid-luteal phases of the menstrual cycle. We had 9 women. We were giving them ovulation kits, doing all the timing. I think we were 9 for 9 in the early follicular phase. Thank goodness, that's not hard to do. I think we were 7 for 9 in the late follicular phase, and we were, I think, 3 or 4 for 9 in the mid-luteal phase. Now, as it turned out, some of those women, when we looked at their hormones later, they were actually in the late follicular phase, so we were able to dump them into that pool of data. But it becomes very hard to get those data. We are working with companies to try to sort of give us more direct and quick answers of what's your hormonal milieu today, and then go and have the test or not. In terms of remnant, yeah, I truly don't know. One of the things I keep on, I feel like I'm answering all these questions, but I don't know. But one of the things I keep on thinking about is the latency of these sympathetic responses. And if you look at the FMD literature, not all studies find that FMD increases in the late follicular phase of the menstrual cycle, which is really counterintuitive if you think about the most well-established effects of estrogen are as a vasodilator that upregulates enos, which is critical to mediating that FMD response. So that's why I start thinking about what is the time course that we might expect with these estrogen responses. And if you manage to test someone right in the middle of the late follicular phase, should we be surprised if we don't see that FMD increases in that phase? I am really dying to do sort of a version of that day study and just kind of like look at what is the direct impact of progesterone. What is the direct impact of estrogen? The one problem with estrogen is obviously that you can't give it unopposed for a prolonged period of time. So we got away with 7 days in the PCOS study in New Haven. But yeah, it's a really, really interesting question. It's one of the reasons that we need the animal studies to sort of rely on, but then with that really quick rodent menstrual cycle, it becomes hard. So yeah.
Well, thank you. That was great. That's kind of where we're at too, is if you don't want to look at progesterone, you could study that peak of estradiol, but that's only a day or two. And is that really enough to see any sort of vascular changes?
Exactly. Yeah. Problem.
Yes. So next question from Jennifer Harper. Do you remember what dose of ethanol estradiol was used in the chlamydia dilation study? And on that tagline, was there also progesterone in that?
No.
Okay.
So that was unopposed, which is why we only did it for 7 days. I want to say it was 30 micrograms per day for 7 days. I don't know if it's weird that I know that off by heart.
No, that's perfect. Thank you. And then
You can skip mine, Dane, because I know we're short on time. Should the students get their questions?
okay. Well, one was somewhat inadvertently answered, but so Talia asked, do you know if any research has been done regarding cardiovascular risk in transgender individuals? Specifically, do male to female individuals have decreased risk of cardiovascular disease if they are on hormone replacement therapy? Or do female to male individuals have increased cardiovascular risk?
Yeah, so I can tell you that with the latter, yes. The former, I actually don't know. I'm sure I've seen it in the presentations. I'll tell you, I got kind of like I said, I got really excited about research in transgender individuals and sort of like with the idea of using it as an opportunity to study the direct effects of hormones. Like I got really excited about PCOS in the first place. And then I went to, it was the Society for Sex Hormones conference where I now cannot remember where it was. And it really, Organization for the Study of Sex Difference, there we go. And it really opened my eyes to how complicated research in transgender populations is. There were a lot of really great speakers who talked about, those different, the impact of timing, which sort of like harkens back to the menopausal data. It really seems that if you let someone's cardiovascular system go with or without certain hormones for enough time, that that matters and that impacts the system somehow. That you can't just drop hormones on board at any time and see the effects you think it's going to. It matters what the vascular system has gotten used to. I have no idea how that works, but it seems to really matter. And that was the take-home message I took away from the current research in transgender individuals. And my suggestion for anyone interested in going down that road would be to definitely hook up with a clinician who's got a big sample size of individuals from whom you can recruit, because I think, would want to very carefully pick the populations that you would study.
Perfect, thank you. So I think we'll probably end with this next question, which is Tom's. And he's curious about receptor expression or quantification, specifically in PCOS in regards to estrogen or testosterone. Maybe not so much the message, but the receptor it binds to as well, if that's been done.
Like what the concentrations of estrogen and testosterone receptors are?
Or just alterations, yeah.
Yeah. For some reason, that just sent me down a path of insulin receptors. Can I quickly answer a question that nobody asked, which is to say, just for all the insulin people, I wanted to say that there's a hypothesis that PCOS results because of a defect in the insulin receptor, that when insulin binds to the insulin receptor, the downstream pathways are activated differently in PCOS. So there's a problem with that insulin receptor, and that's fundamental to PCOS. There's actually people that are arguing that you need to include insulin resistance in the diagnostic criteria for PCOS. So then this downstream problem after the insulin receptor activation causes hyperinsulinemia. And because you have insulin receptors on the ovaries, that causes the ovaries to manufacture testosterone at an increased rate. The testosterone increases adiposity, which further exacerbates insulin resistance, and so on. Dane, I did not answer your question, but that was the one thing I wanted to remember to say about insulin resistance. I had to figure about that loop in here. So insulin resistance people should really care about PCOS, or at least PCOS has made me really care about insulin resistance. I honestly, I hate to say this, I don't know. So the idea behind PCOS and estrogen is that in general, estrogen levels are fairly constant across, because they generally don't have this regular menstrual cycle. So their estrogen levels are sort of like normal-ish. And then you kind of get into the women who are cycling versus not cycling. So it's difficult to come up with one concrete answer to that, because if you have the phenotype where you are cycling and ovulating, all of these hormones are going to look a little bit different. There is crosstalk between the receptors, I can tell you, from my time in the cell lab. But other than that, I don't know, and I don't know if people know what happens to these the density of these hormones in women with PCOS.
Well, awesome. Thank you so much. I really can't thank you enough.
If there's any question, no problem.
Yeah. If everyone could unmute and give a round of applause, it'd be awesome.
Thank you.
Thank
We
you very
called it
much.
too long, but
Thank you.
Thank you for coming, and thank you for inviting me. This was a real pleasure.
thank
Yeah,
you.
I know I've said it like 6 times, but sincerely, thank you again.
No problem.
And all the time too.